This is Chapter 22, Module 3, Lymphocytes and the Adaptive Immune System. The learning objectives of this module are 1. Discuss the types of T cells and their roles in the immune response, and 2. Discuss the mechanisms of B cell activation and differentiation. Lymphocytes account for 20 to 30% of the circulating white blood cells. 3 classes of lymphocytes that circulate in the body are the T cell, the B cell, and the NK or natural killer cell. The T cell is considered thymus-dependent because it matures in the thymus before being released into circulation. The B cell was named not for the bone marrow, where they were produced, but for an organ in a bird where they were discovered. They were discovered in the bursa of Fabricius, which is an organ found only in birds. But for our purposes, you can remember B cell for bone marrow, where it is produced in humans. Most lymphocytes are T cells. The primary types of T cells are the cytotoxic T cells, the memory T cells, the helper T cells, and the suppressor T cells. Cytotoxic T cells are involved in direct cellular attack. They are the main cells involved in cellular immunity or cell-mediated immunity. This type of immunity protects the body when a pathogen has entered into one of the body's normal cells. There are other types of T cells than these 4, but we won't focus on them in this segment. B cells are cells that can differentiate into cells called plasma cells that secrete antibodies. These antibodies are proteins that are known as immunoglobulins. The B cells are responsible for antibody-mediated immunity, which protects the body when a pathogen is in the body fluids, such as the interstitial fluids and the plasma. This antibody-mediated immunity is known as the humoral immunity. Humoral means body fluids. Antibodies bind to specific chemical targets called antigens. Most antigens are pathogens or parts of pathogens or some other foreign compounds. Antigens are usually proteins. The binding of the antibody to its target antigen starts a reaction which leads to the destruction of the cell carrying the antigen. Natural killer cells are large and granular lymphocytes. They attack foreign cells. They also attack normal cells infected with viruses and cancer. They continually police the peripheral tissues, which is called immunological surveillance. The various lymphocytes are not evenly distributed throughout the body. For example, the T cells outnumber the B cells in the body by a ratio of 8 to 1. Lymphocytes can be found in the blood, the bone marrow, the spleen, the thymus, and the peripheral lymphoid tissues. The lymphocytes in these places are visitors, not permanent residents. All 3 types of lymphocytes wander through the tissues, and then they enter blood vessels or lymphatic vessels for transport. T cells move much more quickly than B cells. A wandering T cell may spend about 30 minutes in the blood, 5 to 6 hours in the spleen, and 15 to 20 hours in a lymph node. B cells, which are responsible for antibody production, move much more slowly. A typical B cell will spend about 30 hours in a lymph node before moving on. Lymphocytes typically live a long time. About 80% survive 4 years, and some last 20 years or more. Normal lymphocyte levels are maintained throughout your life because your body produces new lymphocytes in the red bone marrow and the lymphoid tissues. Lymphocytes are produced in a process called lymphopoiesis. Lymphopoiesis involves the red bone marrow, the thymus, and the peripheral lymphoid tissues. This process begins in the red bone marrow, where a stem cell called a hemocytoblast divides to become a lymphoid stem cell. These lymphoid stem cells will then differentiate into all types of lymphocytes. The red bone marrow produces 2 distinct populations of lymphoid stem cells. One group of lymphoid stem cells remains in the red bone marrow and will divide to produce immature B cells and natural killer cells. As these immature B cells and natural killer cells mature, they will enter into the bloodstream and migrate to peripheral tissues. Most of the B cells move into the lymph nodes, the spleen, or other lymphoid tissues. The natural killer cells patrol the body, moving through peripheral tissues in search of abnormal cells. Another group of lymphoid stem cells will leave the red bone marrow and migrate to the thymus, where these cells will mature into T cells under the influence of thymus hormones. The lymphoid stem cells divide repeatedly, producing the various kinds of T cells. As these T cells mature, most will re-enter the bloodstream and return to the red bone marrow. The other maturing T cells will travel to the peripheral tissues, including lymphoid tissues and the spleen. The T cells and B cells that migrate from their sites of origin retain their ability to divide and will produce daughter cells of the same type. For example, a B cell that divides will produce other B cells, not T cells or natural killer cells. This ability to divide and increase in numbers is crucial to the success of the immune system. The lymphocytes generally ignore the body's own cells unless they become abnormal in some way. Natural killer cells are responsible for recognizing and destroying abnormal cells when they appear in the peripheral tissues. This constant monitoring of normal tissues by natural killer cells is called immunological surveillance. Natural killer cells recognize an abnormal cell by detecting antigens on the surface of the abnormal cell. The abnormal cells contain antigens on their cell membranes that are not found on the membranes of normal cells. The natural killer cells will respond to a variety of abnormal antigens and are not selective to specific antigens. In other words, they will attack any cell that contains an abnormal antigen and is not selective to a specific antigen. For example, a single natural killer cell will attack bacteria in the interstitial fluids, body cells infected with viruses, or cancer cells. They are non-selective in what they will attack. If it looks abnormal, they will attack it. Natural killer cells respond much more rapidly than T cells or B cells. While the activation of T cells and B cells is a complex and time-consuming series of events, the natural killer cells respond immediately on contact with an abnormal cell. The T cells and B cells are more selective on their attack of pathogens. We call this adaptive immunity. Adaptive immunity is not present at birth, but must be developed after a person has been exposed to that specific antigen. The process of adaptive immunity is very complex, and for this segment we will only describe the role of the T cells and the B cells, and will not describe in detail the complex process of adaptive immunity. The 4 types of T cells have different roles in the immune response. The cytotoxic T cells are responsible for cell-mediated immunity. They will directly attack cells that have been infected with pathogens. They attack these cells both chemically and physically. The memory T cells are T cells that have already been exposed to an antigen in the past. They clone themselves to produce identical lymphocytes that have the ability to attack the pathogens containing the abnormal antigens. The helper T cells can stimulate both the T cells and the B cells. They are vital to the immune response because they must activate the B cells before the B cells can start producing antibodies. In persons with AIDS, the helper T cell count is greatly decreased and is mostly responsible for the loss of immunity in AIDS patients. Suppressor T cells inhibit the T cells and the B cells to help moderate the immune response. Before an immune response can begin, T cells must be activated by being exposed to an antigen. This activation seldom occurs through direct interaction between a T cell and the antigen. In other words, the T cells don't become activated right away when a pathogen enters the body's tissues. Instead, they only become activated when the normal cells either engulf these pathogens or become infected with these antigens. The cells that engulf these pathogens are the phagocytes. Once the phagocytes have engulfed the antigen, or once a normal cell has become infected, they will present the antigen on the surface of their cell membrane. This presentation of the antigen will attract the T cells. The T cells will then bind to the presenting antigen and become an activated T cell. These activated T cells will divide to become one of the 4 types of T cells: cytotoxic, memory, helper, or suppressor, depending on which T cell type was originally activated. The activated T cells can then perform their designated function. B cells, on the other hand, become activated when they are exposed to an antigen in the interstitial fluid. Once activated, these B cells are on standby, and they must meet with an activated helper T cell before they can divide. Once they do meet with an activated helper T cell, the B cells will divide rapidly to produce plasma cells. The plasma cells will then rapidly produce antibodies that have the same target as the original B cell. The antibodies attack these target antigens to destroy them. When the B cells are activated, they will typically divide several times. They produce either plasma cells or memory B cells. The memory B cells perform similar to the memory T cells. That is, they don't respond to a threat on the first exposure, but on a second exposure, they will divide and differentiate into plasma cells that will secrete antibodies in massive quantities at a very rapid rate. In this way, the body can launch a much quicker immune response the second time it is exposed to a pathogen. This ends Chapter 22, Module 3, Lymphocytes and the Adaptive Immune System.